Hey everybody, in this video we are going to talk about the Ames test. Ames test. Okay, so what is the Ames test? Well, so this is a test that, researchers would use if they are trying to determine if a certain chemical or compound is mutagenic. So let's take chemical X. Okay, now question is this mutagenic? Does it cause mutations in DNA? So what's an example? How about a new preservative for food? So you're a researcher or you're at a big biotech company and you think you've discovered a new preservative and you believe it's totally safe and food lasts forever if you put this preservative into it, and it's harmful to, say, prevents growth of microorganisms, but completely safe to humans. How do we know? How do we know if it's safe? How do we know it's not mutagenic to humans? So, well, we could do an Ames test, right? So there's another question before we get into the test. So, well, maybe that chemical or that new preservative isn't mutagenic on its own, but what happens if it's metabolized, right, inside our bodies? So chemicals are often sent to the liver where they're metabolized. So metabolized by liver enzymes. So what do those products look like? And are those products, after metabolism of the preservative or any chemical, those products themselves could be mutagenic. So what if that food preservative, if we eat it, ingest it, shipped to our liver, metabolized by liver enzymes into some products we don't really know what they are, but are those products mutagenic? So the Ames test can look at all these different things. So, okay, so how does this work? Well, I've never actually done one of these tests, but they're pretty interesting. So the standard bacterium is a Salmonella strain. Salmonella, what the heck is that? So Salmonella typhimurium. Haven't heard of that one before. But, okay, so we're using strains of Salmonella in this test, and we're going to use one or a couple different strains in particular. So let's say this is the wild type strain right here, and the strain is His plus, so it's just a wild type strain. And let's take a look at the sequence of that His gene. I'm going to make a sequence up because it's the concept that's important. So I'm going to put a couple different codons in here for this His gene, theoretical His gene. And I'll diagram the complementary strand. And these dots are to indicate, it goes on. I'm just showing a little part of the gene here. Now let's say these are codons. And if you looked in a genetic code, you would see this code's for methionine. This one, CAG, is glutamine. Glutamine starts, the one-letter code for glutamine is a Q. P and a Q again. So P for proline. Now, let's say this is the wild type histidine gene, one of the genes required to make the amino acid histidine. Well, with the Ames test, one of the strains used in the Ames test is mutated in a gene required to make histidine. So let's say it's this one. Now, let's say the mutation is right here. So instead of a C, let's say there's a T. You put the complementary strand in. Okay, so this is mutated here. And if we look at the codons now, you can see this codon is okay, this one's okay, it's the same. But this one now, because of this point mutation right here, if we looked in a genetic code, table of the genetic code, we see it's a serine now. So proline is changed to serine. Now, let's say this point mutation here makes this protein encoded by this gene. We're going to call the allele the His minus allele. His minus, so this is the His plus wild type, this one's the His minus. So this mutation here makes the protein encoded by this allele non-functional. So it's a null allele. So this is the strain that we're going to use in our imaginary Ames test right here. Okay, so we're going to use this His minus strain. And what we're going to do is take a culture of this bacterium here, the His minus mutant of this bacterium, and we're going to spread it on 2 petri dishes. So let me see if I can diagram 2 petri dishes here. Not too bad. You know, I should probably just print out some pieces of paper with some petri dishes already diagrammed, already printed out, already drawn on the paper and make this process a little faster. Okay, 2 petri dishes. Now, containing the semi-solid medium, semi-solid, what do we say that? Like Jello is semi-solid. So this is medium with agarose in there, and that makes a semi-solid medium. So essentially any petri dish with solid medium, it's not completely solid, right? It's a semi-solid. It's like Jello, gelatin. So the medium in here has the test compound that we are interested in testing to see if it's mutagenic. And down here, this is medium with no compound. So this is going to be our control plate down here. Now, both of these plates have just a little bit of histidine. Say this had histidine, right? The medium had histidine in there because this bacterium right here with a mutation in this histidine, it can't grow unless we add histidine. But these media over here, they don't have histidine. They have just a little bit. So when we plate out the cells onto the medium here, they can grow like maybe 1, 2, 3 cell division cycles. Go through those, divide 3 times. Let's say that. But they'll stop after that unless they undergo a mutation that reverts this T to a C, right? So let's say we plate the bacteria from this culture on here, and we let the plates incubate, and we come back the next day, and we see, oh, okay, there are 3 colonies here on this control plate. So what that means is, the few cells, cells are spread all over here, but in these locations here, one of the cells underwent a spontaneous mutation where that T was switched to a C. So we're going to call that a reversion mutation. We're going to call these revertants. Revertants. So 3 revertants on this plate. Now, the chemical wasn't here, right? So these are spontaneous mutations, right? We talked about ways to classify mutations. They could be induced or spontaneous. Spontaneous mutations, they just occur because of mistakes that just occur in the cell naturally. They're due to just the cell just doing whatever it does. Sometimes polymerase can make mistakes. Sometimes the cell has its own reactive oxygen species that can cause mutations. So there's a basic low level of mutation that just occurs in cells, and it's not due to any chemical in the environment. So we know, we see here that the mutation rate is relatively low here. right? There's only 3 colonies. That's like we plated millions of bacteria here, and only 3 of them reverted to the His plus strain that can grow without histidine in the medium. Remember, there's only a tiny bit of histidine in this media here, just enough to go through 1 or 2 cell division cycles, and just enough. And why do we want that to happen? Well, we want to give the cells time to, you know, undergo a mutation, give them time to revert to prototrophy, histidine prototrophy. So, okay, now let's look over here. Now, let's say in this plate we have, you know, many more colonies. Let's say, I don't know, 100. So this is where we'd have to get into statistics, right? We have to do replicates and compare the numbers here to know that there's significantly more colonies on this petri dish than down here, or under this treatment than under this treatment. And just by looking at these, you can say, oh yeah, okay. So this test compound, it's, you know, definitely causing more mutations, you know, in the bacterium than we would get without the test compound. So we'd say, yeah, test compound is mutagenic. Now, okay, so what did we talked about the possibility that maybe the compound is not mutagenic on its own, but maybe if it is metabolized in the liver, it's metabolized into products that are mutagenic. So for that, we would just need to, you know, I think what scientists do is they mix this, or they treat the compound with rat liver enzymes. I don't know why they use rat liver enzymes, but the rat liver enzymes then can metabolize the product or the compound, and then you can test those metabolites in the same sort of test to see if they are mutagenic. Now, so this type of mutation right here, this is great for seeing if the test compound induces point mutations. Now, we can also use different strains of salmonella. This isn't a bacterium we talk about much in this class, huh? Salmonella typhimurium. We can use different strains of this that have mutations in a histidine gene. Maybe it's even the same one required for production of histidine, right? Because you probably need multiple genes required to make histidine. But there can be different mutations, and we can check if a chemical is more likely to cause a point mutation or an insertion mutation or a deletion mutation by using different strains. So let's say this strain right here is good for point mutations, right? We're looking at the ability of the compound to change this T back to C and make that histidine allele functional. So we can also use a different strain. Now, let's say, I wonder if I can save some time. Hope I didn't go off camera there. Okay. So let's say this is the wild type His sequence. So the other we can use another strain that looks something like this. So I'm going to put A here, T here, G here. You know, but right here, I'm going to skip that C. I'm going to put a space there. There wouldn't normally be a space, right? But I just want to show that this His allele in this different tester strain has undergone a deletion right here. So we're missing this base, or this nucleotide, in the histidine gene here. And that's going to cause a frameshift mutation. So this is a good review because we just talked about frameshift mutations in one of the last videos. So here's a codon right here, and this has been deleted, right? So that shifts the reading frame. So now we have a codon here, and then I would need to know what these bases are here to predict what this codon's going to code for. But if we looked at a genetic code, we could see that this one here is going to code for serine, and this one right here is going to code for arginine, and so on and so forth. So we've shifted the reading frame. So in order for this strain to grow on a medium lacking histidine, what we need to happen is we need a mutation that inserts a base somewhere around here to reestablish the reading frame, the normal reading frame, and then give us the normal sequence of, or to allow an amino acid with the correct histidine sequence and to allow that protein to be functional. So I hope that makes sense. So we can use this type of strain right here that has a deletion in the histidine gene to test if a chemical is mutagenic, but mutagenic in a way that is more likely to cause deletions or insertion mutations. So, and how would we reestablish the correct reading frame here? Well, we could delete 2 more nucleotides, right? And maybe this protein would still be functional, lacking 1 amino acid. There's a chance that could happen. Or inserting 1 nucleotide, that would reestablish the correct reading frame. Okay, so long story short, we can use different strains of Salmonella, different strains, say 1 strain to test for point mutation, let's say reversions, and to test indel, which is insertion or deletion. Indel mutations. Indel mutation reversions. Okay, so we can have 2 different types of strains to test the ability of chemicals to cause point mutations or to cause insertion and deletion type mutations. So one more thing on the Ames test. So found an interesting paper, and I think there are hundreds of these interesting papers. And so what would scientists use the Ames test for? So, dropped something, sorry. So take a look at this paper, Comparative Studies. It is just an abstract. You can get it online. Comparative Studies of Mutagenicity of Urine from Smokers and Non-Smokers on a Controlled Non-Mutagenic Diet. Crazy, huh? So what did they do? They aimed to determine whether the differences in urinary mutagenicity between smokers and non-smokers result from increased urinary mutagenicity from dietary heterocyclic amine mutagens in smokers compared with non-smokers. Did I repeat that line twice? I may have. So essentially what they're doing is they are checking urine from smokers and non-smokers and looking at diet, right? Because there's this idea that, diet, especially individuals who eat a lot of fried meat, and fried meat results in heterocyclic amines, which are mutagens. So essentially they're going to check the urine from smokers and non-smokers with the Ames test. And I believe it says Ames test in there somewhere. Yeah, Ames test. See right here. So what they did was they concentrated the urine using some sort of resin, and they checked for mutagenic activity in the Ames test. So the urine of smokers was significantly more mutagenic than that of non-smokers when on both the raw, boiled, and the Western diets. And you can take a look at the abstracts, pause it, and read more about what that means. But it's pretty interesting. I thought, I'd never really thought about that before. If you're a smoker, your urine contains mutagens, or higher levels of mutagens than the urine of non-smokers. Crazy. So a lot of useful things you can do with the Ames test. And I think in the next video, we are going to take a look at a couple DNA repair process, or at least one double, a DNA repair process that repairs double strand breaks. Okay, see you in the next video.